pathway by which a chemical element circulates through and is recycled by ecosystem is known as biogeochemical cycling. So the terms to remember are: elements can be recycled, or they can accumulate in reservoirs or sinks. Reservoirs or sinks are important because they can be held for long periods of time and can replenish the supply if required, or for elements that are detrimental, for example CO2. Oceans can become sinks for CO2, where they can be kept for thousands of years away from the atmosphere. So a simple example to understand biogeochemical cycling, we can look at the water cycle, where we focus on the processes of evaporation, condensation, and precipitation. So if you look at the water cycle, reservoirs or sinks are in glaciers and ice and snow, also as groundwater storage, and also water storage in oceans. We can also have freshwater storage in lakes. So these are sinks and reservoirs that can replenish the supply if required. Let's look at the nitrogen cycle. For nitrogen cycle, the major reservoir is the atmosphere. 78% is nitrogen gas. However, the nitrogen gas is not usable, and unless it is fixed into a usable form, for example as nitrites or ammonium. So nitrogen fixation biologically is by bacteria via the enzyme nitrogenase. N-fixing bacteria can be in the form of free-living bacteria, for example Azotobacter, or symbiotic bacteria, example Rhizobium in nodules. Other than biological fixation, now there is about 30% of fixed nitrogen that's manufactured in chemical plants, especially for fertilizer. So for natural fixation, other than bacteria, and fixation by lightning can also occur. Other than N fixation, the main processes that is involved in nitrogen cycle are, for example, nitrification, where nitrogen is oxidized to ammonium, then nitrite, and then nitrate. Important bacteria here are Nitrosomonas and Nitrobacter. It's important because nitrate is the form that is usable by plants. When plants and other organisms die, decomposition occurs. Decomposition by bacteria is also known as ammonification because ammonia is released during the decomposition process. So now we have learned N fixation, nitrification, and ammonification. Under anaerobic conditions, a process that is a reverse of nitrification or denitrification can occur when nitrate is reduced to nitrogen. This is carried out mainly by bacteria, Pseudomonas, Clostridium, and Bacillus, for example, which used nitrate as electron acceptor. The electron accepting power of an element is measured by its redox potential, where the more positive the reduction potential, the greater the species affinity for electrons and the greater the tendency to be reduced. So here we can look at the redox potential of the common processes that you can observe in the environment. So aerobic respiration has the highest redox potential and is the preferred step by most organisms. Next in line is denitrification or nitrate reduction, where nitrate supplies the electrons. Lower down the redox potential, we have manganese reduction, iron reduction, sulfate reduction, and also the carbon dioxide reduction to methane or methanogenesis. So in an environment where possible, aerobic respiration will occur first, and when the oxygen level decreases to anaerobic levels, other processes can begin to occur. If nitrate is available, nitrate reduction occurs. Next, manganese, then iron, and finally methanogenesis. So this chart shows the end cycle where ammonification occurs by decomposers, and the ammonium that is formed undergoes nitrification to nitrite and then nitrate. Then nitrate is also again assimilated by plants, and plants is consumed by animals. So for nitrogen fixation, we see here examples of symbiotic nitrogen fixing bacteria living in the legume root nodules, for example rhizobium, and also free living nitrogen fixing bacteria, for example Azotobacter. Here we see the nitrogen cycle according to its oxidation state from positive 5 to negative 3. So here we have ammonification or catabolism, where the organic nitrogen is degraded or decomposed to ammonia. This ammonium can be assimilated or undergo nitrification to nitrite and nitrate. So the bacteria that is involved here are Nitrosomonas and Nitrobacter. When the conditions are suitable, for example when the oxygen levels decrease, denitrification can occur, where nitrate is used as an electron donor by bacteria such as Bacillus and Pseudomonas. And for atmospheric nitrogen gas, nitrogen fixation can be carried out by free living bacteria, example Azotobacter, Cyanobacteria, and also Clostridium. So in the past, the ammonium oxidation from here to here is only known to occur when the so nitrification is a aerobic process. However, now we know there is anaerobic ammonium oxidation, which means ammonia can also be oxidized in anaerobic states. So this is observed first in wastewater treatment plants, where they were able to isolate bacteria that can carry out the falling process, where ammonium and nitrite reacts, forming nitrogen gas and water. So this ammonium oxidation from ammonium to nitrogen gas is carried out by the first and foremost example is Brocadia anemoxidans. Now other bacteria are also known to carry out anemox reactions. So here we look at the global nitrogen cycle. What is important is a lot of nitrogen gas in the atmosphere, and the fixation is biological, lightning, and also industry. So industry now is becoming more and more important than lightning. So whatever that is fixed is assimilated by plants, and when they die, ammonification and nitrification occurs. So there is a transfer from rivers to the sea, but mainly the processes are the same. Whatever that is not recycled is sedimented, into rocks and sediments, and this can be a form of reservoir or sink. Other than nitrogen cycle, let's look at the sulfur cycle. Sulfur is released from rocks through weathering. So weathering processes, for example, changes in heat and also rain, will help release sulfur from the rocks, and upon exposure to air, the sulfur is oxidized to sulfate, and this sulfate can be assimilated by microbes and plants. Relative to nitrogen, sulfate is less important because if you remember, nitrogen is assimilated and used in enzymes, proteins, amino acids, DNA, RNA, whereas for sulfate it is utilized mainly in certain amino acids. Sulfate reduction is actually quite low in its redox potential. For the sulfur cycle, we have facultative sulfur reducers, but we also have obligate sulfur reducers. So some bacteria needs to use sulfur, whereas others will only use it if nitrogen or other elements are not available. So sulfur is used by sulfur reducing bacteria to get their energy, where it reduces sulfur to hydrogen sulfide, and this is done in combination with oxidation of acetate, succinate, and other organic compounds. Some of the bacteria involved in the sulfur cycle are extremophiles, and most of them are in the domain archaea and living in hot springs and thermal vents, where there is usually a higher concentration of sulfur. So for the sulfur or sulfate reduction, there are two main pathways. One is assimilatory sulfate reduction, and the other is dissimilatory sulfate reduction. So the main difference is assimilatory sulfate reduction is where the bacteria reduces the sulfate in order to synthesize sulfur containing cell components. Therefore, the term assimilatory. Therefore, the amount required is actually in small, but for dissimilatory sulfate reduction, the sulfate reduction bacteria reduces the sulfate in large amounts in order to obtain energy, and expelling the resulting waste as hydrogen sulfide. So these are the two main sulfur reduction pathways, one assimilatory and the other dissimilatory. Sulfur oxidation occurs both in aerobic pathway and also anaerobic conditions, where the aerobic sulfur oxidation is mainly carried out by phototrophic bacteria. For example, similar to the ones that you see in the Winogradsky column. Here you can see the sulfur cycle, which shows the key reactions and the sulfur species. So you have sulfide oxidation by phototrophs, okay, and you have sulfate reduction, both dissimilatory and also assimilatory. So in the aerobic conditions where you have ample oxygen and nitrate, hydrogen sulfide and sulfur are thermodynamically unstable and is used as electron donors for ATP production by chemolithotrophic microbes. Okay, for example, Thiobacilliaceae in terrestrial and Beggiatoceae in aquatic environments. So the phototrophs that you see that utilizes hydrogen sulfide as electron donors to fix carbon dioxide, the phototrophic sulfide reduction cycle pathway here is mainly carried out by the purple sulfur bacteria in Chromathiales. So this is the bacteria that you see in the Vinogradsky column. That is also why you see sulfur granules in the bacteria. So in oxygen-rich conditions, sulfate is generated as it is more stable, and the sulfate can act as an electron acceptor. So here sulfate is reduced, dissimilatory and also assimilatory pathway. For the global sulfur cycle, we can see the emissions from waterlogged soils, for example mangroves and paddy fields, mainly in the form of hydrogen sulfide, and then artificial emissions from the burning of fossil fuel, mainly in the form of sulfur dioxide. And when it rains, you have wet deposition, and sometimes you have dry deposition from dust, mainly in the form of sulfate. So whatever that is not recycled is usually sedimented back into the crust, Earth's crust, and it is from the weathering of the Earth crust that we obtain the sulfur here. So other than the sulfur cycle, we can also see sulfur in DMSP production. So dimethyl sulfonylpropionate, that is produced by algae. So this is a normal exudate that is released by the algae in order to maintain cellular osmotic balance in the sea. So as the algae photosynthesize, as the algae grows, DMSP is released. And this DMSP is used by bacteria that has DMSP lyase that breaks DMSP down into dimethyl sulfur and also acrylate. So acrylate is a food for the bacteria, and DMS is volatile gas that is released into the atmosphere. So when DMS is released into the atmosphere, sometimes it is photooxidized by the sun rays into sulfate. And sulfate is known as a cloud condensation nucleus, where the availability of sulfate allows cloud formation. There is a hypothesis that mentions when global temperature increases, tropical algae, which releases more DMSP, can increase its distribution to wider areas, and therefore the amount of DMSP release is also increased, which will bring about more cloud formation. And this could be a feedback mechanism to reduce global warming. Okay, let's look at the carbon cycle. So the main processes are photosynthesis, where carbon dioxide is fixed into biomass, and also respiration, where carbon dioxide is released. Anthropogenic impacts from human activities, where we have increased CO2 emission from fossil fuel burning. We will also have methane production from agriculture and also livestock. So this also contributes to the carbon cycle, because both CO2 and methane has global warming potential. So human emissions are important because it has grown so large that our activities has brought about a change in the climate. So trees are important because whatever that is fixed can reduce the amount of CO2 in the atmosphere. And soil carbon that is fossilized can remain in the soil for up to 10,000 years as a sink and reservoir. So that is also where the problem arises, because humans extract fossil fuel, coal, and other products from the soil, for burning, for production, and this increases the amount of CO2 in our atmosphere, and that has brought about the climate problem that we are now facing. And this is another picture showing the carbon cycle, where we have processes in orange and the reservoirs in purple. So this also shows the carbon cycle, where atmosphere has the CO2 and we have reservoirs in the form of dissolved organic carbon and also sediments. So these reservoirs are important because they can be sinks and reservoirs for carbon for thousands of years. So because of the increasing CO2 in the atmosphere, we have seen a concurrent increase in global temperatures. And we can see the effects of warming, where the melting ice in the North Pole has seen a reduction in the ice cover during summer. This figure is from 2007. So now ships can actually pass through the North Pole safely during summer. We can see the disappearance of glacier from Europe, and also the disappearance of snow and ice from Mount Kilimanjaro in Africa. Other than increasing temperatures, we are also seeing rising sea levels, longer periods of drought that has dried up lakes and also caused a loss of habitat. Finally, let's look at microbiologically induced corrosion in petroleum industry. For microbial corrosion, the formation of biofilms are essential, where you can see flagellated microbes or microbes that are motile. Once they are attached, they lose their motility and produces extra polysaccharide that forms the biofilms. So biofilms are important because they allow the bacteria to attach better and also prevent the bacteria from toxins or other elements that are detrimental to the bacteria. However, once the biofilm are thick enough, either the biofilm is sloughed off or the bacteria reverts back to its motile mode and moves on to form a new biofilm. Microbiologically induced corrosion is a serious problem in the petroleum industry, where holes and cracks can be formed by bacteria. We can see the effects of bacteria where sulfite concentrations are high. That means there is sulfate reducing bacteria that reduces sulfate and releases hydrogen sulfide. However, when biocides are released into the piping system, you can see that the amount of sulfide reduces. Then when biocide treatment is stopped, the biocide concentration decreases, while the bacteria starts sulfate reduction again, and you can see sulfide increases again. So the corrosion reaction is dependent upon sulfate reducing bacteria. So here we see sulfate reducing prokaryotes that reduces the sulfate in the fuel into H2S. The H2S reacts with the metal to form rust and also gas, hydrogen gas. So this hydrogen gas can actually reacts with CO2 to form methane via methanogens, which are also present in petroleum. So both these processes are important in the microbiologically induced corrosion of metal pipes. So other than microbiologically induced corrosion, microbial utilization of hydrocarbons are also of interest. The term used are hydrocarbon utilizing microorganisms, or humbugs. These are of interest because of the use of this bacteria as a way to bioremediate oil spills. So at the moment we have about 250 kinds of bacteria that can live in fuel. Ecological study of this bacteria can help optimize the bioremediation step, which means we are able to use these microbes to degrade oil spills faster and into less dangerous bioproducts. Most of it uses Pseudomonas aeruginosa. So this bacteria is highly adaptable and metabolically very diverse, and is able to degrade many types of waste. So we can see the carbon cycle, where the carbon dioxide in the atmosphere can dissolve into the water, and through this buffering potential, employing bicarbonates and carbonates. So other than the buffering potential here, biological activity that fixes CO2 into biomass, and after the organism dies, there is decomposition, and then sedimentation and burial, and further deep burial into crust and rocks. And these are only brought out back into the atmosphere through volcanic activity and outgassing.